I'd like to thank the Academy organizers for inviting me to speak today at the Interdisciplinary Symposium on To See or Not to See, Unbiased Answers to Forensic Questions. Today, I'll be talking about eliminating bias in forensic algorithms and statistics. In this talk, I will be referring to cybergenetics' TrueAllele probabilistic genotyping system, which was used to analyze the DNA evidence in the case example. There are both targeted and untargeted approaches to examining evidence. Let's begin by looking at targeted approaches in forensic science. In 2004, fingerprints taken from the Madrid bombing were compared with an exemplar fingerprint from an American citizen, Brandon Mayfield. We see the latent evidence print on the left and the reference target on the right. 3 different fingerprint examiners said that there was a match between these 2 fingerprints. However, there wasn't. It was the wrong person. Forensic science involves making comparisons between items, typically an evidence item and some other item, often a target. But there are issues with these comparisons. For example, when comparing with a target, to what extent are alternatives considered of other targets? Evidence and targets are not compared directly. Patterns taken from the evidence are compared with patterns taken from the target. For example, the fingerprints in the Madrid bombing were taken from a bag of detonators. These patterns can have tremendous complexity, so usually the pattern as a whole can't be compared with another pattern. Instead, there are features that model the data, and the features of the evidence are compared with the features of the target. This comparison introduces other issues. For example, if you have a feature and there are different values of the feature, then there's uncertainty as to which values in the evidence are more probable. And on the target, there are now choices to make of what the feature values would be, as well as the targets. In statistical science, a likelihood function is used to assess these different choices. The likelihood is the probability of observing the evidence data given a hypothesis or assumptions about what the data might contain. The data is typically an evidence pattern in forensics, whereas the hypothesis chooses a target, which we'll call T, and it's written in shorthand as the probability of D given H, or the conditional probability of the data given the hypothesis. Likelihood measures how well an hypothesis explains the data. Also, a better probability model with better math and more variables can give a more accurate likelihood value. With targeted forensic approaches, the likelihood centers attention on the target T, and it asks how well does this choice of the target explain the evidence. Targeted approaches run the risk of introducing human bias. Forensic comparison often involves making choices. There's the choosing of which evidence items to analyze. There are the data features to focus on when examining evidence. There's the choice of a target person, specifically in a targeted method, and then there are decision criteria as to how to conduct the comparison and how do we know that there's a match. Making definite choices can restrict forensic possibilities and cause error. It can lead to scientists overstating their conclusions as well as ignoring other possible answers. Better interpretation methods do not limit choices. Instead, they consider all the alternatives. Confirmation bias is a well-known problem in forensic science. With choices comes the risk of confirmation bias. Dr. Raymond Nickerson of Tufts University, in his classic article of 20 years ago on confirmation bias, defined the phenomenon as an unwitting selectivity in the acquisition and use of evidence. Unwitting because people don't do this consciously. Selective in that people choose which data to acquire and which evidence to use in their analysis. In his article on confirmation bias, Nickerson talks about hypothesis-driven thought, the hazards of having a hypothesis determine what information you're seeking and how to interpret it. Here are 5 topics that he covers. One is restricting one's attention to a favored hypothesis, a target. Secondly, preferentially treating evidence that supports one target over another, existing beliefs. Thirdly, looking only or primarily for positive cases and not using negative results to help rule out what isn't true. Fourth, overweighting positive confirmatory results by focusing more on what's supporting your argument than what might be against it. And finally, seeing what one is looking for in the evidence instead of what's actually there. Here are illustrations of confirmation bias taken from the case that we'll be discussing later. When someone focuses on a target, you can think of that as a defect in the likelihood function. When we're looking at the probability of the data given the hypothesis, there's undue emphasis on some fixed target hypothesis, H. The image shows a comparison of strands of hair under a microscope. On the left, we see the target, the defendant in the case, and on the right, there's evidence of hair that's taken from a garbage bag. Looking at these 2 strands of hair under the microscope, they look very similar. Are they the same? Are they not? Well, from the picture, maybe it's a match. But by focusing on the target, we're not asking, what does non-target hair look like? What would a comparison of someone else's hair look like who wasn't a target of the prosecution with the hair evidence that was found? This short tandem repeat, or STR, DNA data shows another type of confirmation bias where the examiner might change the data based on the target. We see that this in the likelihood function, probability of the data given the hypothesis, where the data is altered with preferential treatment of evidence that supports the target. The figure shows an electropharogram, an EPG. On the x-axis, we see the different alleles separated by size, 15, 16, 17, and 18. Vertically, we see the relative fluorescent units, which is measuring how much of each allele has been amplified in the polymerase chain reaction, or PCR. Looking at the data, we see 4 allele peaks of different heights. The 4 different alleles can be combined into 10 different pairs of alleles, or genotype values, as shown on the right, 15, 15, 15, 16, and so on. In this case, there's a target, say a 17, 17, that would be formed by the homozygous combination of that tall 17 peak allele with itself. Without changing the data, that 17-17 homozygote allele pair is just one of 10 possibilities and might not indicate strong statistical support for the target. But some DNA methods apply thresholds that discard lower-level data, and these data thresholds can artificially discard exculpatory evidence. Here, we see a threshold of 50 RFU being applied, and the peak at 18 on the bottom right is now gone. There are only 3 alleles. Well, that leaves the allele peaks 15, 16, and 17. And now there are only 6, not 10, possible genotype values, as we see on the right, and that 1717 option, that's the target, becomes more prominent. Raising the arbitrary threshold higher to 100 RFU removes even more data that points away from the target. Now both alleles 16 and 18 are gone. There are only 2 alleles left, 15 and 17, and there are only 3 possible pairings of genotype values as shown on the right. Now the 1717 target looks even more likely, being one of 3 possibilities. Raising the threshold even higher to 150 RFU would now get rid of 3 of the 4 alleles, 15, 16, 18, all under 150, all gone. All that's left is the 17 allele, which leaves only one genotype possibility, the homozygote 1717. It looks like a virtual certainty that it's the target if the data are changed to this extent. Fewer alternatives, going from 10 alternatives to just 1, inflate the target's match statistic, and can produce apparently strong DNA evidence to the wrong person. Another variant of confirmation bias looks primarily at positive cases. Here, the likelihood function is placing an undue emphasis on positive data, indicated with the D+ probability of the data given a hypothesis. The example from the case is a P30 test for semen. On the right, we see how this test is read. A negative result only has a red control band, whereas a positive result has both a red control band as well as a red test band. On the left, we see the data from a first set of P30 tests, which the lab called as weakly positive for the middle test on item 16. Let's zoom in on that CT control test region for the middle test on item 16. On the right, there's a blue arrow that's pointing to where the band might be. Do you see the band? Do you see it faintly? Do you see it at all? Well, if you were looking primarily for positive case results, that might bias you to see a band, whereas a more neutral assessment might not. Focusing on positive results might lead one to have more belief in positive outcomes than in negative outcomes. On the left, we see the first set of semen tests that were taken at the hospital, which may have been weakly positive. On the right, for the second set of tests that were done at autopsy, those semen tests were entirely negative, and the data were not shown to the jury at trial. There was an emphasis on looking primarily for positive results. A fourth confirmation bias scenario overweights positive confirmatory instances. Here, on the top right, we see the probability function itself is changed, where there's an undue emphasis of positive over negative results. The data from the case we're looking at is a search for semen using a microscope. With semen, there may be very many cells present, dozens and dozens of cells that would indicate with a Christmas tree stain, red sperm heads and green flagella. What we see is one red cell, and it might be semen, it might not be semen, it might be sperm, it could be yeast. And even if it's sperm, as a great poet once said, how many cells must an analyst see before they can call it semen? Our last confirmation bias example is seeing the the target that someone is looking for, instead of what might actually be there. On the top right, we see the likelihood expression, the probability of the data given the hypothesis H. Here, I've emphasized the conditional given, because if the focus is on what you're looking for, you're not really conditioning on the hypothesis for the data as much as knowing what you want from the target. In the example case, as shown in the central brown column, DNA was taken from the victim's body, from his face, scrotum, rectum, fingernails, and on the right, we see how these different items would support the prosecution theory, with facial bruising, scrotal bruising, anal rectal injuries, and that a victim fought back. And on the left, we see the DNA from the target defendant. His DNA wasn't found on the face or scrotum, but it was found on the rectum and on the fingernails, supporting the prosecutor's theory. In the DNA analysis of the many items in this case, there was DNA found from unknown people. On the right, we see that DNA from an unknown person was compared with the DNA from the victim's body. And for this unknown person, we see yes, there is a DNA association between this unknown, never identified individual with the DNA from the victim's body. That is stronger support for what the prosecutor wants to argue. Stronger than the no, yes on the left column from the DNA from the target defendant. The non-target alternative on the right better explains the prosecutor's theory. But when you're seeing the target, you're only seeing what you're looking for, and the focus remained on the defendant instead of perhaps looking for this unknown person. Targeted analysis can lead to confirmation bias. We'll now look at some non-targeted approaches to practicing forensic science. The key is to consider all the alternatives. Now the likelihood function we want is the probability of the data given an alternative hypothesis. An alternative hypothesis, written as not H, can ask, what if it's someone else, someone who's not the target? And that's abbreviated here as the probability of D given not H. When considering the non-targets, how well do they explain the evidence? That's precisely what the likelihood of the alternatives tell us. The likelihood ratio formalizes this balance between both hypotheses. It lets us evaluate the evidence data under both the target hypothesis H and the non-targeted complement alternative hypothesis, not H. So we have two hypotheses, H and not H. H is focused on the target, not H is focused on the alternatives. These two hypotheses are mutually exclusive, which means they don't overlap, and exhaustive, which means between the target and the alternatives, they cover all the situations. In his confirmation bias article, Professor Nickerson explains why this property is important. He wrote that an observation gives one little evidence about the probability of the truth of a hypothesis, unless the probability of that observation, given that the hypothesis is true, that is the likelihood, is either substantially larger or substantially smaller than the probability of that observation, given that the hypothesis is false, the likelihood of the alternative hypothesis. There are different ways of writing down the likelihood ratio. Some formulas are better for explaining what it means, others are better for calculating. Likelihoods are computed from the variables used in forensic models. A variable is an uncertain quantity that can take multiple values. The probability form of the likelihood ratio is ideal for overcoming confirmation bias. It can be derived using Bayes' theorem. This form is the ratio of posterior probability divided by prior probability. The posterior Q tells us the probability of a target after we've examined the evidence. The prior P gives the probability of the target before we see the evidence. The probability ratio of posterior Q divided by prior P computes the likelihood ratio. A proof that these equivalent forms all give the same likelihood ratio numbers can be found in the paper below. The probability form of the likelihood ratio provides a non-targeted approach to weighing evidence. First, calculate probabilities without ever looking at the target. To do this, we need to find the prior probability, P, for all the different values that can occur before we ever look at the evidence data. This prior probability is usually determined in forensics from population prevalence studies. We also need to calculate the posterior probability Q for all possible values based on the evidence data. So we have a computer examine the evidence data, and from that, it can produce these posterior probabilities after having seen evidence. These two probability functions, the prior and the posterior, are for all possible values. They're non-targeted. They don't involve the target. They've never seen the target. That comes next. In step 2, after having determined the prior and posterior probability functions for every possible value, simply plug in the target value. Here we see that the likelihood ratio of the target is just that ratio of probabilities, posterior Q, evaluated at T, divided by the prior probability, also evaluated at target T. This non-targeted approach lets us decouple data analysis from comparison. They're done in 2 different steps. Let's look at an example involving DNA evidence. DNA mixtures arise when 2 or more people leave their DNA on the same evidence item. Let's continue with the 4-person mixture data example that we looked at earlier. We'll take a look at calculating a likelihood for a DNA mixture. Superimposed on the 4 data peaks of 15, 16, 17, and 18, we see an explanation that adds together different genotypes. In blue, the large amount of 15, 17. In orange, the 16, 17 allele pair. In green, a 16, 18 allele pair. And in red, a small amount of a 15, 18 allele pair. When those 4 contributor genotype values are all added together, we get different heights of predicted amounts of alleles. And adding them together, looking at the tops of those added togethers, those stacked alleles, we see a pattern that's very similar to the underlying data pattern. So those 4 genotypes explain the peak pattern, giving a better explanation of the observed data and therefore a higher likelihood. When a computer is analyzing DNA evidence, it doesn't just look at this one explanation, it considers all possible genotypes and combinations in different amounts and other second-order statistical effects as well. And when it's done, it can produce a complete analysis of the probability function Q. Here we see a picture of the posterior genotype probability function Q. It's for one of the 4 contributors at one of the 20 genetic loci. The computer has separated out this evidence genotype and determining it up to probability. It's pulled it out of the mixture data using statistical computation. Of the roughly 100 possible allele pair values, on the horizontal x-axis we see 8 of those allele pairs. Vertically, we see their probabilities. the number on top of each of the bars in this bar chart gives the probability after having seen the data, the posterior genotype probability for this contributor at this genetic locus based on the mixture data. To calculate likelihood ratios, we also have to consider the prior genotype probability. That's shown in brown. The function p is indicated by the brown bars that are sitting right next to the blue bars. All of these genotype possibilities have been considered without any bias. There's no target involved in this calculation. The target is not known to the computer. Only after all that groundwork has been done do we calculate a likelihood ratio at the target. So we have our posterior genotype probability Q, our prior genotype probability P. Now we set a target, say the 15-17 allele pair. We focus only on the target, and this is done only after we finished analyzing the evidence. Circled in red around the 15-17 allele pair, shown with the oval, there's a ratio of the blue bar's posterior probability of 48% divided by the prior probability of 6%. That ratio of 48 divided by 6, which equals 8, is the likelihood ratio. So the likelihood ratio using this approach focuses attention at the very end just on the target, but it could have calculated a likelihood ratio for any of the allele pair values that we see here, or even ones that we don't see here. You just plug them into the formula, and out pops the likelihood ratio at this locus, the 15-17 allele pair. It's equal to 8, calculated in an unbiased, untargeted way. This two-step approach gives us an objective way of calculating a likelihood ratio without focusing on a target. It's untargeted forensic science. The approach is objective because it never sees the target, and it's unbiased. It does it in 2 steps. The LR calculation is split into 2 parts. The first part never sees the target but does all the heavy lifting. This approach also eliminates direct comparison between patterns from the evidence and from a target. It works with data features instead, and necessarily involves probability because it's complete. It's not making choices. It uses all of the STR genotyping data. It considers all of the alternative genotypes, and it gives the results probabilities. It's a non-targeted probabilistic approach. Probability is introduced, but so is more accuracy, so is more reliability, and bias is eliminated. The target is unknown when examining evidence data. We first described this in the paper below over 10 years ago. This 2-step unbiased approach can eliminate bias in other forensic subdisciplines as well. Let's now look at a DNA case example, contrasting targeted versus non-targeted analysis of evidence. The example case is California versus Lopez. This case was resolved last year. A young man was accused of rape and murder of his girlfriend's toddler son. He was facing the death penalty or life in prison if convicted. The very young child was 2 years and 10 months old. There were bruises to his face, his genitals, his rectum, and an autopsy showed brain swelling, skull fracture, cheek bruises, and asphyxia. A rectal swab from the boy showed semen, and the swab matched the defendant's DNA. There were 2 different views of the case. The lawyers on opposing sides had different theories or hypotheses about what happened. The prosecution believed that the defendant raped and killed a 2-year-old boy who lived in his house. The defense believed that an abused toddler died. The defendant had nothing to do with his death. A hair was found on a garbage bag that contained clothing from the deceased child. Was it the defendant's? Wasn't it the defendant's? Well, looking at it under a microscope, a forensic analyst looked at the target together with the evidence and believed that this hair was unique in her experience, and this was a match. Comparing the strands of hair under a microscope produced a match of the target and evidence hair. DNA analysis was done on the hair root. Probabilistic genotyping computer analysis was done on the STR data in a non-targeted, unbiased way. After first calculating prior and posterior probabilities for every possible genotype value, comparison was made with the 6 references shown in the first column. Shown in order, these are the victim's sister, his brother, his mother, the defendant, his brother, and the victim himself. The 6 red X's indicate that no statistical association was found between the hair root and any of these 6 individuals. The defendant and the square next to him is highlighted in red, so you can see the results in a more targeted way. The 4 rows on the right are for 4 unknown individuals whose DNA genotypes were found at the crime scene. The red X's indicate there was no match to any of these individuals either. An important question in the case was whether the defendant's semen was present in the child's rectum. Rectal swabs were taken first in the hospital and then later at autopsy. As we saw earlier, the hospital P30 results shown on the left were weakly positive, whereas the later P30 results taken at autopsy were entirely negative. Cybergenetics' subsequent probabilistic genotyping computer analysis of the crime lab's STR data confirmed these results. Comparison of the non-targeted analysis with the 10 reference items shown on the right produced no matches except for the defendant. His match statistic was a number in the thousands, a logarithm of 3, and so we see in blue the match statistic of the log-LR 3.81 associating the defendant's DNA with the hospital rectal swabs. Cybergenetics also confirmed the crime lab's results for the rectal swabs taken at the autopsy later on. The defendant is not associated with these second set of rectal swabs, nor is anyone else except for the victim. Another forensic question was whether the defendant's sperm was on the victim's penis. From the microscopy analysis, 1 cell was seen. By some protocols, this would mean that there's no strong support for sperm being present. Other protocols might find that sperm is present. How many cells must be seen to call it semen? And whose cell is it? Here again, DNA analysis can be helpful. Untargeted, unbiased computer examination of the penile swab sperm fraction produced genotypes. These genotypes were compared with all 10 references. Except for the victim, the autosomal STR analysis produced no matches. A YSTR analysis done by the crime lab was weakly positive. There was a lot of forensic DNA evidence in this case. The crime lab analyzed 97 evidence items using STRs. They reported on 43 matches, and they discovered 1 unknown person. Cybergenetics processed 77 STR data items using our probabilistic genotyping TrueAllele software. We reported on 138 matches that we found, and we discovered 5 unknown people in the evidence. An independent crime laboratory using TrueAllele confirmed our findings. There's a reason why targeted manual review of DNA mixtures produces so little information. The main match statistic, the combined probability of inclusion, is very uninformative. It doesn't use peak heights, it applies thresholds. Basically, it just counts up how many loci an analyst reported. It's not correlated with information. As described in a research paper based on crime lab data, published 5 years ago, as the title states, inclusion probability for DNA mixtures is a subjective, one-sided match statistic unrelated to identification information. Untargeted, unbiased DNA computer review of mixtures, on the other hand, has been extensively validated and shown to be highly reliable. Shown here are 2 validation papers published in the Journal of Forensic Sciences, one from 2015, where we studied DNA mixtures containing up to 5 unknown contributors, and another validation study extending these TrueAllele interpretation results to DNA mixtures containing up to 10 unknown contributors. The next few slides compare how much identification information the crime lab obtained using its manual review DNA mixture methods relative to TrueAllele's automated computer interpretation. The first 3 charts we'll see are from the county crime lab and show the amount of match information, and the second 3 charts show the same comparisons but now with TrueAllele's probabilistic genotyping results. The legend on the right shows the degrees of match association. At the bottom in gray is just inclusion, and then proceeding in groups of 6 orders of magnitude, we have a range that goes from 1 to 6 log units of likelihood ratio, then up to 12, 18, 24, and 30. These correspond to numbers like 1,000,000, 1,000,000,000,000, and so on. This is a DNA match table. The items of evidence are shown in the rows listed on the left. The references that they're compared with are shown across the columns above. There are 6 known references, sister, brother, mother, defendant, brother, and victim, and 6 unknown references on the right half of the chart. Almost all of the evidence items are DNA mixtures. Comparing these 25 items with the references, we see that the crime lab was able to identify the defendant, shown with some light blue match statistics in 3 rows, as well as identify the victim through inclusion with the gray bars in the victim column. When the crime lab compared the next group of 25 mixture items with the 6 references, again, they mainly found the defendant and the victim, as indicated by the blue and gray highlighted cells. The crime lab's targeted manual methods are not finding DNA associations between the evidence items and any of the other 4 known references. Continuing with the last group of 25 evidence items, again we see associations mainly with the defendant and victim. Here there is one association with an unknown female found on one of the evidence items. It's easy to see from this crime lab analysis why the prosecution thought that they had their man. All the DNA seems to be pointing to the defendant and to no one else. Cybergenetics' untargeted computer analysis shows a completely different story. The match associations light up the match table, showing DNA connections between the evidence items and other known individuals as well as unknown individuals. Continuing with the next batch of 25, we again see that the computer found more match results than the crime lab could. If we look at the person in 21A-EC, that's the column right next to the right of the victim's column, we see a number of match associations with evidence items. In fact, more associations to the evidence than even the defendant has. In this last group of 25 evidence items, we see that the computer continues to find matches to most of the references, not just to the defendant and the victim. Noticeably absent, though, is there's been no DNA matches to the mother. The mother is in the third column, just to the left of the defendant. In the 138 DNA match associations to 77 evidence items, not a single match association is for the mother. The computer's information-rich survey of the DNA landscape of this crime scene left us with two strange puzzles. First, where's mom's DNA? Lots of different people left lots of DNA, but the primary caretaker left none. Secondly, there's a conflict between the two rectal DNA results. Why was the defendant's DNA found in the initial hospital rectal swabs, but not later at the autopsy? There are two opposing views of the same DNA evidence, reaching diametrically opposed conclusions. The prosecution, relying on what the crime lab found, mainly the victim and the defendant being present on evidence items, believes that the forensic evidence shows that the defendant raped and killed the 2-year-old boy who lived in his house. The defense is looking more at the computer's larger results that finds far more matches. In their view, an abused toddler died. The forensic evidence shows that the defendant had nothing to do with his death. Perhaps this is an example of confirmation bias. Assume a hypothesis, conclude the hypothesis. Assume the alternative, conclude the alternative. The prosecution looks at the evidence and they see crime. In their view, the forensics proves guilt. The defendant's DNA is everywhere. It's on the child's clothing. It's on a vibrator. It's on fingernail clippings. They see it on hair. They see a rectal swab. They see it on the boy's body. What they see is the defendant's DNA. The defense looks at the same evidence, and they see no criminal. They see lots of people's DNA in a messy, dirty, spermy house. The crime scene is littered with debris and dirt and clutter and lots of people's DNA all over everything. In their view, nothing specifically points to the defendant. The clue to the first puzzle, where's mom's DNA, is in her statements and testimony. Question, did you and the defendant have sex that night? Mother, just a blowjob. Question, did he ejaculate inside your mouth? Answer, yeah. Question, what did you do after that? Answer, went and cleaned myself off. Question, what did you clean yourself up with? Answer, baby wipes. Question, and then what did you do with the baby wipes afterwards? Answer, threw them away. Question, where did you throw the baby wipes away? Answer, I don't remember if I threw them in the trash can in my bedroom or if I threw them in the restroom trash. So the mother's DNA was masked because her hands were covered in the defendant's semen. The child was in toilet training. The child's primary caregiver had the defendant's semen on her hands, swamping out her own DNA as she attended to her child, which is why forensic science couldn't detect her DNA. The mother was spreading the defendant's semen from her hands, 2 baby wipes, 2 garbage bags, and whatever she touched, like to the body of her child. There was no probative value in this expected DNA. The defendant's DNA arrived through transfer. And now the key to the second conundrum. Why was the defendant's DNA on the rectal swabs in the hospital, but not later on at the autopsy? The answer is in the pathologist's testimony. Question, and in this particular investigation at the hospital, did you collect DNA swabbing from the victim's anus? The pathologist replies, yes, sir. Question, and how did you do that? Answer, the same way we did for his genitals. Swabs were broken from a sterile package, Sterile water was applied. I would place the swabs around the skin of the anus in a circular manner. Insert 1 inch to 2 inches, pull them out, and then they would have been handed off to law enforcement. Question. Okay, so you never stuck the swab into the rectum or into the anus more than 2 inches? Answer. Right. The beginning part of the anus and the rectum is sufficient. So the answer is in the rectal swab that cleaned the anal area. First, at the hospital, the mother had transferred the defendant's semen from her hands to the toddler's bottom. The first pathologist swabbed the boy's anus, cleaning the external anal semen onto the swab. Before the swab was rectally inserted, the so-called rectal swab already contained the external anal semen. Later on at the autopsy, the toddler's bottom had been cleaned by the pathologist at the first hospital swabbing. There was no more external anal semen to collect, and there never was any internal rectal semen. So the second, quote, rectal swab was devoid of DNA. There was no external semen to collect. This evidence is based on misleading terminology. It was an anal rectal swab, not a rectal swab. And the procedure cleaned the anal region, collecting the semen from the external part of the boy. There was never any internal semen. There were two competing worldviews of this case. There was lots of forensic DNA evidence, and there was confirmation bias from both sides. But ultimately, there was a final verdict. Yes, the prosecution was target-driven, focused solely on the defendant. The defense was non-target-driven. They believed that other explanations were better than that the defendant had raped and murdered the child. And many forensic experts educated the jury. But ultimately, the non-targeted scenario better explained the forensic evidence. The jury listened, understood, and acquitted the defendant of all charges. Instead of ending up on death row without better, untargeted forensic science, the defendant was set free. And as a postscript to this case, given the incredible disparity between what the prosecution truly believed based on the evidence that they had in front of them from their lab, and what the jury ultimately rendered as a verdict, the county no longer seeks the death penalty. Let's wrap up by looking at the impact of non-targeted forensic science methods on criminal justice. As this presentation has shown, it's possible to eliminate much human bias from forensic science. Confirmation bias is real, it's prevalent, and it's dangerous. Targeted forensics can introduce confirmation bias by focusing the evidence in a particular way on a particular suspect. Likelihood ratios help with confirmation bias because they consider both targets and non-targets. I also showed how the likelihood ratio can be calculated without targeting suspects. This is done by splitting the calculation into two parts. In the first step, what's needed is to examine the evidence for the probability distributions P and Q, the prior and the posterior probabilities, and get those probability functions for all possible untargeted values. Every possible genotype, for example, in the case of DNA, without ever seeing, looking at, or knowing what the target is. That's a lot of work. It takes computers. but it can be done, and it is done. And secondly, after all that heavy lifting is done, after step 1 is finished, then any target value can be plugged into those probability functions to calculate the likelihood ratio in an untargeted, unbiased way. It could be done for one defendant, for 10 suspects, or for an entire database of 15 million offenders on CODIS. This talk has also shown the role of unbiased methods in forensic science. It was a limited DNA analysis that targeted Defendant Lopez. It was the limited forensic science using manual methods that were based on biased targeting that led to his prosecution. He faced the death penalty because of forensic limitations, and it was only better, untargeted, unbiased computer methods that ultimately saved him. The Lopez case is just the tip of a very large forensic iceberg. Innocent people need better forensic methods. They need untargeted methods. You can't establish that somebody's DNA is not present in evidence if the methods begin with establishing inclusion. How do you prove exclusion if your starting point is a targeted inclusion? Victims of crime need better forensics. Targeted methods limit what DNA evidence is capable of. Informative computers can reach in to complex mixtures, separate them out in an unbiased, untargeted way, and afterwards make comparisons that objectively inculpate those individuals who left their DNA and exculpate those people who didn't. Finally, past, present, and future cases need accurate, objective, automated solution. We need to open the past, use CODIS to review cases from 5, 10, 20 years ago using newer computer methods that might be unfamiliar to CODIS but are much more powerful at giving untargeted answers. We need to be using the best untargeted methods possible in current cases so that defendants like Mr. Lopez don't face the death penalty because of limited forensic analysis. And going forward in the future, this should be an expectation for all fields of forensic science, using accurate and objective computers in an automated way to recover all the identification information from forensic evidence. I would like to thank the American Academy, particularly Chris Milroy and Agnes Winokur, for inviting me to speak at this interdisciplinary symposium. Many people worked very hard on the Lopez case at Cybergenetics, including William Allen, but particularly Jennifer Bracamontes, who also testified in the case. And a special thank you to public defenders Kelly Kulik and Mike Ogle, who saw the power of confronting limited forensic science with more powerful forensic methods. If you'd like to contact me about this talk or anything related to it, my email address and website URL are shown. Thank you very much.